In this video, we will learn about antibiotics. These are chemical substances, that is a protein produced by microorganisms to kill or inhibit the growth of other microorganism. We are just exploiting this property of killing the other organism or microbes or restricting its growth. So the term antibiotic was coined by Selman Waxman in 1942. Anti means against, biotic means living. Generally, it means a bacteria. So these are obtained from certain living organisms such as bacteria, yeast, molds, etc. These days they are produced on a larger scale using certain microbes also. Now if we see who discovered the first antibiotic, the first antibiotic named penicillin was discovered by chance by a scientist, that is Alexander Fleming, in 1929. So we can see the picture of Alexander Fleming here. So how did we discover by chance is Fleming was working in London hospital. So he was culturing bacteria on a agar jelly in petri dishes. So petri dishes are these glass bowls. Some of these plates became contaminated by colonies of a blue-green mold known as Penicillium notatum. Now, Fleming noticed that there was an area of clear jelly around each mold colony, so where there was no bacteria growing. So, penicillium stopped the growth of bacteria. So, later he extracted the chemical released by this green mold and found that this chemical is preventing the bacteria to grow. So here we can see this is the soaked with concentrated penicillin, and around it has a large area where it is very clear. So bacterial growth, which is abundant white dots, which will disappear because the penicillin was incorporated. Now clear zones around the mold colonies growing on a plate of bacteria can be seen. Now Fleming found that the substance could be extracted from this mold and could be prevented the growth of the bacteria, and such extract he named it as penicillin. So this penicillin proved to be a wonder drug during the World War. So it was used for treating infectious human diseases in 1940. So in particular, it yielded very good results in treating gonorrhea, a sexually transmitted disease. So thereafter, a whole lot of antibiotics such as streptomycin, chloromycetin, aureomycin, ampicillin, etc. have been discovered. Now these are effectively used against many infectious diseases. Now if we take the sources of antibiotic, there are many microbes that act as sources. Example, we have penicillin. Source is a fungi known as Penicillium chrysogenum. Next we have streptomycin, where the bacteria Streptomyces griseus will give the chemical. Erythromycin is extracted from another species of Streptomyces, that is Erythrius. It is a bacteria. Neomycin is extracted from Streptomyces freidae. This is also a bacteria. Then we have aureomycin extracted from Streptomyces aureofaciens. Terramycin extracted from Streptomyces ramosus. We have other antibiotics like cephalosporin extracted from Cephalosporium aeromonium fungus. We have chloramphenicol, which is extracted from Streptomyces venezuelae. Now there are antibiotics which are broad spectrum antibiotics and narrow spectrum antibiotics. So the antibiotics which act on a variety of pathogenic microbes are termed as broad spectrum. That means these are not specific for bacteria, they just prevent the growth of most of it. Example include chloromycetin, aureomycin, terramycin, etc. Now if we have a narrow spectrum, that means these are against a specific pathogenic microbes. Example include penicillin, erythromycin, streptomycin, etc. If we come to uses of antibiotics, so these are medicines against various disease causing microbes. It prevents the spoilage of food, etc., especially meat and flesh, and helps in preservation. Treating animal feed to prevent internal infection. It can be used in antiseptic creams. It can also be used in controlling plant pathogen. I hope this topic is clear to you. Thank you.